Click for IB Biology. In this presentation, we're looking at the muscle structure. We'll look at the muscle fibers, the individual muscle cells called myocytes, and we'll begin our study of the force generation through the contraction of the muscle. There's a PDF down in the description that you can use to annotate yourself with your own notes. There's a quick quiz with answers so you can check your understanding. And then at the end of the video, there are links to other videos in this series as we go into more and more detail. So we're looking at what you might call the growth structure of the muscles. At either end of a muscle, you will get a structure called a tendon. And now tendon is connecting muscle to bone. Contrast that with ligament, which is bone to bone. And then we have the bulk of the muscle structure in the center, like this. And then it turns out that if we look at the internal organization of this large muscle structure, it's made up, as we go in with finer and finer detail, certainly visible to the eye are what we would call muscle bundles. Groups of muscle cells organized together, bound by collagen. Muscle cells don't run the entire length of the muscle. They tend to be in sections, and those sections are also connected with collagen for the efficient transfer of the force of contraction. And so we have these bundles of muscles with the individual muscle cells called myocytes in there. And this is an image here of the individual myocyte. If I just move to the next diagram, the myocyte, myo being muscle, cyte being cell. First thing is that they are multicellulated. I beg your pardon, multinucleated structures, and within them are what are called myofibrils. Now, myofibrils are protein molecules that run the entire length of the cell, and these take two forms. One's called actin, one's called myosin, and we will be studying those in more detail in the next videos as we find out how it is that these actin and myosin actually generate the force of contraction, which is what muscles do. Now, around these bundles of protein molecules called myofibrils, so here they are here, there is a complex tubular network. One could think of this as being like the endoplasmic reticulum, but adapted to the use in muscles. And so you can see that its actual name is the Sarcoplasmic reticulum. And in various sections, it might be known as the T-tubules or the lateral sacs. But the important thing to remember here is that stored within these sarcoplasmic reticulum tubulocytes are calcium ions. And this will be a critical link to our study of the mechanism of muscle contraction. And when that muscle contraction occurs, what we know is that it's necessary to have a supply of adenosine triphosphate to create the shortening. And as this shortening process occurs, the length of the muscle reduces, and it is that which creates the force, or the effort, if we use the physics word, which brings about the movement of the lever at the joint. So how is this force actually created? And to find the answer to that, we will need to go in at the microscopic level. This image here is from an electron microscope, and we need to go in at that resolution in order to see the actual structures. In this image, you might note that the cell membrane is here. The muscle cell is running broadly in this direction. So the section of the photograph's like this. So there's lots and lots of mitochondria producing that ATP that we know we need. And then the contraction mechanism will be found through the study of the myofibril proteins that you can see arranged here in a kind of banding pattern going diagonally across our muscle cell here. Links to the process of muscle contraction will be found here. We'll first of all consider the nerve impulse. Then later on we'll go on to look at the internal mechanism things. Playlist is on the right hand side. If you want to subscribe to the site to look at all of our videos, then the button's there as well.